I am Dr.R.Krishnan, Assistant Professor, PG and Research Department Microbiology, St.Joseph College of Arts and Science, Autonomous Cuddalore. Here I am going to present a topic about DNA microarray, its applications. Before going to DNA microarray, so you have to know some basic information about the cell. So it is said to be the basic unit of any living organism. It contains a complete copy of organism's genome. In human, we have trillions of cells. Other organisms like yeast, they are unicellular, a single cell. Cells are of many different types. So like blood cells, skin cells, nerve cells, etc. But all can be traced back to one special cell. So like the fertilized egg cells. So this is the structure of an eukaryotic cell. So next is some basic differences between the prokaryotic cell and eukaryotic cells. In case of prokaryotic cell, lack a distinct membrane-bound nucleus. Example, in bacteria. In the second type of cell called a eukaryotic cell, they possess a distinct membrane-bound nucleus, and they are larger and more complex in structure than prokaryotic cell. Example, mammalians and yeast cells. So if you see the eukaryotic cell, the nucleus, they are membrane enclosed structure which contains chromosomes. Then DNA molecule carrying genes essential to cellular function. And the cytoplasm, it is nothing but the material between the nuclear and cell membrane, which includes fluid, a cytosol, and organelles and various membranes. And for the very important organ for protein translation, they are nothing but ribosomes. So they are said to be a small particle composed of RNAs and proteins that function in protein biosynthesis. And other organelles of this eukaryotic cell, so like membrane enclosed structures found in the cytoplasm. In a vesicles is nothing but a small cavity or sac which is containing a fluid. And then they have a mitochondria, so found in most eukaryotic cell, in which respiration and energy generation occurs. And the mitochondria also having the genetic material DNA, which code for certain proteins. And another important organelle called is centrioles. They have a pair of cylindrical bodies which is composed of microtubules, so which actually determines the cell polarity and very important during mitosis and meiosis process. And then they also having another organ called endoplasmic reticulum. It's nothing but a network of membranous vesicles, so to which the ribosomes can be often attached. And next is the Golgi apparatus. It is also a vesicle which is very important for the translation. And the cilia is nothing but a membrane hair-like projection which is found on the certain type of cells, so which helps this eukaryotic cell, a microorganism, can able to move. And this is the chromosome of a eukaryotic cell. Chromosomes, images of chromosomes. So let's say about the chromosomes. In human genome, we have 23 pairs of chromosomes. Out of this 23 pairs, 22 they are said to be the autosomal pairs, and the 23rd, the final pair, is called sex chromosomes. In female, it is called XX, and male is called XY. And in each pair, one chromosome is paternally inherited and other is maternally inherited. Chromosomes are made up of a compressed and confined DNA. A protein coding gene. A gene is a segment of a chromosomal DNA that directs the synthesis of a protein. And next is the cell division of eukaryotic cell. So normally they will undergo a process called mitosis. First, nothing but the nuclear division produces two daughter diploid nucleoid acid or nucleic identical to the parent nucleus. After mitosis, the chromosome undergo meiosis. That is, a two successive nuclear division produces four daughter haploid nuclei different from original cells, which leads to the formation of gametes. And this is the process of mitosis happens on the chromosome. So it involves four different phases, that is called prophase, a metaphase, anaphase, and telophase. And finally have here two daughter cells. And this is called as a meiosis stage. And they also undergo a process called as a recombination method, where the interchange of the chromosomal DNA within the same chromosomes. And next we're going to see some of the concepts about the DNA molecule. DNA, they can be expanded as a deoxyribonucleic acid, or DNA molecule is a double stranded polymer composed of four basic molecular units called nucleotides. Each nucleotide consists of a phosphate group, a deoxyribose sugar, and one of the four nitrogen bases, adenine, guanine, cytosine, and thymine. And the two chains of the DNA strand are held together by a hydrogen bond between the nitrogenous bases. So according to the rules, guanine pass with the cytosine and adenine pass with the thymine. And this is the double helical structure of DNA. So the DNA replication process. Normally the DNA can replicate by either by conservative or dispersive or semi-conservative mode of replication. So next is the genetic and physical maps. So physical distance is the number of base pairs. Genetic distance is expected the number of crossovers between two loci, and then per chromatide or per meiosis measured in morgans or in centimorgans. And this is the maps of genetic and physical. And let's come to the human genome in numbers. So 23 pairs of chromosome. So it will be approximately 3 trillion base pairs. And the number of genes will be 30,000 to 40,000. It's an approximate value of the genes. And second important after DNA, the biomolecule will be the protein. So nothing but a large molecule composed of one or more chains of amino acids. So these amino acids, it is a very important molecule present on the protein, which makes the protein structure. So there are 20 different organic compounds containing a basic amino groups and then carboxyl group. The order of the amino acids is determined by the base sequence of the nucleotide in the gene coding for the protein. Example, hormone, enzyme, antibodies. And this is the structure of 20 different amino acids, as you know that. And this is the protein structure. It is involved in the translation process. So it happens on the ribosomes, where the amino acids are joined together and form a polypeptide chains. So this is the different structures of the protein: primary, secondary, tertiary, and quaternary structures. And this is a different cell device, prokaryotic cell with an example and eukaryotic cell with an examples. So next is very important topic called the central dogma of molecular biology. Here, the expression of the genetic information stored in the DNA molecule occurs in two different stages. So nothing but one is the transcription, where the DNA is transcribed into RNA, and second is called as the translation, which is the mRNA is translated to a protein. So this is the entire process of central dogma or gene expression process. And the next important nucleic acid called RNA. So they are called ribonucleic acid or RNA molecule is a nucleic acid similar to DNA, but some differences will be there. It is single stranded, having a ribose sugar rather than a deoxyribose sugar, and a uracil rather than thymine. And RNA plays an important role in protein synthesis and other chemical activities of the cell. And there are several types of RNA molecules, including mRNA, tRNA, ribosomal RNA, and other small RNAs. And the very important concept called a genetic code. Here the DNA, they having a sequence of 4 different nucleotides, and protein, they have a sequence of 20 different amino acids. So the corresponding between DNA 4 alphabets and the protein 20 letter alphabet is specified by the genetic code, so which relates nucleotide triplets or codons to amino acids. So this is the genetical table column where each triplet code will specify one specific amino acids. So this determines the amino acid composition. So this genetic code will be read by that is specific tRNA with the help of this anticodon region. After reading, the specific amino acids are attached and form a growing polypeptide chain. And the gene, they are also having a codon and non-codon region. A coding region of the gene is called as exon, and a non-coding region of the gene is called as intron. So during the mRNA processing, the introns are removed by splicing process. So next, the final very important topic called DNA microarray. So far we have seen some basic introduction about molecular biology, the difference between prokaryotic cell and eukaryotic cell, the replication of eukaryotic cells, and transcription, translation, like that. So next is DNA microarray. So normally this type of technology can be used to monitor the amount of gene expressed in a particular cell. So we can also quantitatively measure the concentration of the gene expression and qualitative analysis of particular gene expression. So DNA microarray. So this technology has to be hybridization concept. So it relies on the hybridization property of nucleic acid to monitor DNA or RNA abundance on a genomic scale in different types of cells. So by using a DNA microarray, one can also monitor the routine level of a particular gene inside the cell. And this technology can also use to monitor malignancies, any immunodeficiency problem, genetical disorders, etc. So it is a nucleic acid hybridization method. So where the mRNA will be collected from a particular cell at the particular time of duration, where it will be converted into cDNA and hybridized to microarray chip. So the gene expression analysis can be monitored by different ways, like serial analysis of gene expression technology, one method, and also by using short oligonucleotide arrays, then long nucleotide arrays, then fibric optic arrays, and cDNA arrays. And the application of microarrays. So they can be used to find the mRNA present in the cell. Then genotyping, so estimation of DNA copy number, then determining the identity of descents, then measuring mRNA by decay rates, then identifying protein binding sites, then determining the subcellular location of gene products. So this is the entire mechanism of a DNA microarray, where it starts with the express mRNA collection and conversion of cDNA of the mRNA, then hybridization of cDNA to the molecular probes, where hybridization taking place between the cDNA and then probe attached on the chip. After attachment, it will be read by the scanner. So this is the entire process of that DNA microarray technology, where the hybridization will be taking place between the cDNA and the probe attached on the chip. After hybridization, it shows the emission of a signal which can be monitored in the form of colors. So this is the array instruments. So this is the a microarray chip where the hybridization will be taking place, which can be indicated by colors. So this is the same images. So this is the entire process. And the sample preparation. So when you're going to perform the DNA microarray, get the specific sample, separate the cells, isolate all the expressed mRNA, and convert them into cDNA. So this is the hybridization process. So that is the binding of cDNA target sample to cDNA probes on the slides. So this is the chamber. So where the slide can be kept. This after hybridization, that can be monitored by using a scanning devices, nothing but a detector. And finally, those cells which actually binded to the probe can emit signal, and that can be used to confirm the particular gene has been expressed in a particular cell. So this is the technology. So where they will be inserting the chip into the scanner. So this is the binding. So after binding, it emits a signal at the particular spots that can be read by the scanner. So this is the overall image after hybridization between the cDNA and probes. So this is the entire cycle of this DNA microarray and the whole experiment flow charts. So it starts with experimental design, microarray experiment, image analysis, like that. And this is the references. Some of the references were I just collected for this topic. Thank you.